So we'll break it down for you piece by piece. So first, diabetics based on continuous glucose monitoring data. What we started off with was a collection of blood glucose time series. We got our data from Medtronic, and what this data consisted of was blood sugar readings every 5 minutes, here shown for a 24-hour period. We had 106 real type 1 diabetics, denoted by a 4-letter code. Each diabetic had a little over an average of 60 days of data with 288 readings. So we had a lot of data to work with. And what we wanted to do with this data was cluster the diabetics based on some inherent property that they shared. And the way we went about doing this was using a wavelet-based filter to filter our time series, get filter data that was in a way similar to analyze, and then from that data we got our clustering. An unsupervised learning algorithm, what this means is that we wanted to develop an algorithm such that we started with our time series, put them into the algorithm, got the clustering, and then we go back and look at what the clustering tells us. We started off with no predisposition about our time series, no hypothesis that we were trying to fulfill. Rather, we just wanted to develop this algorithm so that we could start with the time series, get the clustering, and then see what we can learn about the original diabetics.
So the first thing we want to do is introduce you to diabetes and what the disease entails. And there's actually 2 types. There's type 1 and type 2. In our research this summer, we've only been looking at type 1 diabetes. So when we refer to diabetics or diabetes, we're just referring to type 1. And this is an autoimmune disorder in which the body can no longer produce insulin. And insulin is the body hormone responsible for the regulation of blood sugar levels. Without the insulin, glucose, or sugar, remains in the bloodstream, and over time, it can cause serious harm to the body. And estimates show that nearly 1 million Americans have this disease. So without the body's ability to regulate its own blood sugar, diabetics have to constantly monitor it manually using either a finger prick or by wearing a continuous glucose monitor. Based on these readings, they can then determine whether or not they need an artificial injection of insulin. And what we have here is the graph of an actual type 1 diabetic's blood glucose over a 24-hour period. And what we want to do with graphs like this is create a digital or numeric signature, so to speak, that can capture the important characteristics of this graph only in a much more condensed form, and use that signature to compare it to other diabetics. But to be an accurate representation of this time series, we need to factor in these short-term variations, the sudden drop in blood sugar at 3 in the morning, or the sudden surge in blood sugar at noon. And one of the tools that we want to use to do that are wavelets. And these are relatively new mathematical tools developed only in the last 30 years. We can think of them as functions to analyze other functions, or as filters to analyze signals. And a signal is just a sequence of numbers. And what a filter does is it operates on a signal S, and it creates a new signal that emphasizes certain characteristics of that old signal. So the signals we're dealing with are blood glucose time series, and the features we want to emphasize are those short-term variations in blood sugar. So for each wavelet, there's actually a pair of filters associated with it. There's a low-pass filter, denoted H, and a high-pass filter, denoted G. The particular filter coefficients shown here are derived from the D4 Dobeshi's wavelet, which is the wavelet that we use this summer. And we don't want to actually get into the derivation of these coefficients, but we do want to show you how these filters work. So we'll take an example signal S, and in equation 3 and 4, we calculate the results of the low and high-pass filters. To see where these come from, it's basically just a dot product. We take the 4 entries in H, we multiply them component-wise by the first 4 entries in S. Sum the results, and this gives us our first entry in equation 3. To get the second entry, we do the same dot product, only with the 3rd through 6th entries in the signal S. We can similarly calculate the result of the high-pass filter. Now, the actual algorithm we use for computing these results is known as Malaz's pyramid algorithm. You can sort of see why by the way we have things set up on the left. And also notice that we renamed the results of the high and low pass filters. On the left, we have the detail coefficients. These are just the results of the high pass filter. They're called detailed because they pick up the detail or the differences in the signal, those short-term variations. And on the right, we have the approximating coefficients, just the results of the low pass filter. They're approximating in the sense that they're a weighted average of the original signal. Now, after applying the filters one time, we can actually pick up more detail by applying them a second and a third time, this time to the approximating coefficients. Doing so gives us a grand total of 7 detailed coefficients and a final approximating coefficient. What we're interested in is the relationship that these 7 detailed coefficients have with one another and what that can tell us about the original signal.
So when it comes to blood glucose time series, a particular reading isn't just an independent data point. It's going to depend on other readings. So your blood sugar at one time is going to depend on what it was 5 or 10 minutes before that. So the same idea holds after we filter the data. So for example, the value 1.55 is going to depend on the value 9.29. Additionally, the coefficients in different levels are all going to be statistically dependent. The 9.29, 1.55, and 11.88 are all going to have a definite relationship between them. So with this relationship in mind, we calculate 8 statistics from data related to the detail coefficients. First, we calculate the mean, standard deviation, skewness, and kurtosis of the detail coefficient magnitudes, and then we calculate those same 4 statistics for the linear predictor errors. And the linear predictor errors are data related to the detail coefficient magnitudes, and this is where this relationship between the different readings comes into place, and it is crucial with the linear predictor. So the reason we chose this method was that methods similar to this have actually been used successfully before. In 2004, Liu et al. Used them to identify the artist or artists of a particular painting, and the 2005 RU group used them to identify forged handwriting samples. So what we want to do with these statistics is we want to calculate a similarity between 2 diabetics. And this similarity is going to be in the interval 0, 1, with 0 being not similar at all, and 1 being very similar. And this similarity we're going to use in the Laplacian eigenmap method. So the Laplacian eigenmap method is the clustering algorithm we've been doing. The point of the Laplacian eigenmap method is that we start off with k points in Rn, where n is very large, so we can't picture these points graphically. We don't really have a geometric intuition about them. And we want to produce k points in Rn, where n is 1, 2, or 3, so we can represent these points graphically. And the algorithm is centered around a weight matrix W, whose entries indicate how similar 2 points are in that high dimension. So the entries are going to be in that interval 0, 1 that indicates the similarity. And the idea is that we want to keep points close in the higher dimension, close in the lower dimension. The algorithm reduces to finding eigenvectors of this generalized eigenvalue problem, Lz equals lambda dz, where L and D are 2 matrices derived from W. And what we need to do is calculate 2 eigenvectors, z1 and z2, corresponding to the 2 smallest non-zero eigenvalues, lambda 1 and lambda 2. And then using these eigenvectors, we plot the points graphically. So let's recap exactly what our algorithm consists of. We start off with a collection of blood glucose time series we got from Medtronic. Then we use a Wavelet-based filter to get detail coefficients. Then we get some statistics from data related to the detail coefficients. And using these statistics, we calculate a similarity between 2 diabetics. And using this similarity, we put into the Laplacian eigenmap method to get clustering.
Now for each diabetic that we have data for, we want to get a graphical representation in R2, which we obtain by plotting the entries in the eigenvectors associated with the 2 smallest non-zero eigenvalues. When we do that, this is the graph we get. And images like this are fairly consistent with what we've seen in the literature on clustering algorithms, but it's not exactly apparent where the clusters are at in this graph, if there are clusters, and what those clusters mean. So to determine these things, we went back and looked at the statistics for several diabetics for several days. We noticed that 3 statistics in particular, the skewness and kurtosis of the coefficient magnitudes, and the skewness of linear predictor errors varied significantly from one diabetic to the next. So using those 3 factors, we created a classification scheme to which each diabetic would belong to one of 3 groups and associated the color with each group. And when we colored the graph based on that, we obtained these 3 banded clusters. We have the greens in the lower left, the reds in the middle, and the blues in the upper right. And what these banded clusters represent is some inherent relationship in the data. There's something the diabetics colored green have in common, something diabetics colored red have in common, and something diabetics blue have in common. So the next thing was we wanted to determine what this commonality was. And we also wanted to find how do these statistics manifest themselves in the blood glucose graphs. What does having a high kurtosis actually tell us about the blood glucose graph of that diabetic? And that led us to the idea of a PLA, which is a piecewise linear approximation of a function that minimizes the number of pieces necessary to remain within the specified distance of the original function. And this was first mathematically formulated by Stone in 1961, who was trying to fit pieces, linear pieces, to curves. And what we want to do is fit these linear pieces to blood glucose time series. So with the additional criterion that we need to minimize the number of linear pieces and remain within that specified distance at each point where blood glucose is measured. So the purples represent one possibility. And then, using a constant tolerance for each diabetic, we can define the number of segments in the PLA to be the PLA index. So the PLA index is a measure of variability. The higher the PLA index, the more segments, the more variable, as this blood glucose graph on the left is. The lower the PLA, the less variable. And then we can create an average for each diabetic, all of their days, to get a single value, which describes this variability of the diabetic's blood glucose graphs. And again, as before, we can create a classification scheme to which each one belongs and associate a color with that scheme to color our first eigenmap graph. And doing that, we get the same sort of banded appearance. We have these greens in the lower left. It's a little bit different this time. We have the reds in the middle, and we have the blues in the upper right. And so this correspondence between the coloring suggested to us that our wavelet-based clustering algorithm was picking up this PLA, this measure of variability in the blood glucose graphs.
All right, so what exactly did we do? We took a wavelet-based approach to looking at diabetes. We clustered our diabetics based on this wavelet-based approach because the methods have been used successfully before to pick up these inherent similarities between the diabetics. And then we went back and clustered the diabetics based on the PLA index. And these wavelet clusters and these PLA clusters actually corresponded. So this single PLA index, this singular value, is picking up on all the analysis we did with the wavelets. So what we hypothesized is that further research will show that the PLA index can be a very valuable tool in classifying diabetics, helping treat diabetes. So take these two examples again. On the left, we have a high PLA index. This diabetic has a very variable graph. He doesn't know where his blood sugar is going to be from 1 minute to the next. It's very unpredictable, and hence he can't treat his diabetes very well. On the other hand, on the right, we have a low PLA index. Everything is very low lying. There are no sharp jumps. His blood sugar is very predictable, and therefore he can better treat his diabetes. So even if his blood sugar is very high, it's predictable. So there's only small changes that need to be made in order to properly treat it and get the diabetic truly healthy. So we have some acknowledgments. Dr. Aboufadel at the REU, who served as our advisor this summer. Dr. Larry Shep, who provided us with the Medtronic data. Medtronic, who obviously owns the data, and the NSF, who helped fund us. Thank you very much.
Any questions?
Do you happen to know the corner frequency of the filters you were using? You were using a high pass and a low pass filter.
Yes.
But at some point it has to cut off, so it stops passing through numbers. Do you happen to know where that cutoff is?
How many times we applied the high pass filter?
Not exactly, no. Like, is 100 too high or too low? Is 1,000 good enough? Like,
1,000 what? I'm not completely understanding your question.
a high pass filter passes through high values, large values, yes?
The high pass filter is picking up the differences in the values. So there's no threshold as far as what it picks up. It's going to pick up any type of difference. It's not,
the high doesn't deal with both.
yeah, there's not a cutoff as far as what differences it's picking up, and that it throws out, I see what you're saying now, that it throws out all the ones underneath it. Rather, it picks up all these differences and it tells you these differences. We didn't threshold the differences and then look at that. Does that answer your question?
Yes,
Okay.
thank you.
Was there a reason why you picked the two lowest eigenvalues rather than the two highest or any other two?
So it's basically an optimization problem where it boils down to the minimization principle that you need is finding that lowest eigenvalue. And you can actually, so at each eigenvalue where you go up, you sort of lose localization. So the minimum minimizes it, the second one doesn't quite minimize it, but it differentiates, and that's where you get the different dimensions.
Other questions? All right, let's thank our speakers.